Hello, my name is Hannah Nightingale, and today we will be talking about the research study that I chose, which is modeling breast cancer using CRISPR-Cas9 mediated engineering of human breast organoids. It is important to understand the key background information surrounding breast cancer because this is the main focus of the research study. Aside from skin cancer, breast cancer is the most common amongst women in the United States. Breast cancer can begin in different parts of the breast, and the breast is made up of three different parts: the lobules, ducts, and the connective tissue. The lobules are the glands that produce milk. The ducts are the tubes that carry the milk to the nipple. The connective tissue, which mainly consists of fibrous and fatty tissue, surrounds and holds everything together. Most breast cancer does begin within the ducts or lobes. Breast cancer arises from the uncontrolled and accumulated growth of mutant cells residing within the epithelial cells. And it is very extremely common. About 250,000 cases of breast cancer are found in women, and 2,300 cases are found in men annually. Sadly, each year, 42,000 women and 510 men die from breast cancer. So breast cancer arises because organogenesis will take place, which is the transformation of healthy cells into cancer cells due to the chemical and genetic changes that take place. Cell division occurs in an uncontrolled manner, which is very genetically complex, and heterogeneity can be seen. Biological heterogeneity refers to cancers from the same origin site, but between and within tumors, there's a variety of physical, chemical, and genetic profiles. In the figure to the right, it exemplifies a cancer that has the same origin site, but within and between, there's a large amount of diversity, which is why genetically complex cancers, such as breast cancer, are very, very hard to target. Breast cancer is far too common, and it can affect both men and women. The graph formulated by the CDC depicts the annual rates of breast cancer that are diagnosed in women from the years 1999 to 2017. The graph from the top, created by the American Cancer Society, states the probability of a breast cancer diagnosis in women from the ages of 20 to 80 and includes the lifetime risk. So to talk about some statistics a little bit more, approximately 1 in every 8 women will be diagnosed with an invasive form of breast cancer each year. 1 in 39 women will die from breast cancer. And cancer is the second leading cause of death in the United States. And 1 of every 4 deaths is due to cancer. So you may ask, what has been done to decrease these numbers? Well, this research study has been conducted, and it will further be discussed in the rest of this presentation that covers the use of CRISPR-Cas9 to create genetically engineered breast organoids. This research study mainly focuses on the precise initiating events that predispose normal epithelium to neoplasia, because information surrounding this topic is so poorly understood, and there has not been much investigation done in great depth. Also, neoplasia is known as the uncontrolled abnormal growth or cells or tissues in the body. And when that word comes up throughout the presentation, that is its definition. So CRISPR-Cas9 was a vital tool in this study because researchers wanted to see if breast organoids can be generated using this technology. And if so, they would be studying the initiating events and perform experiments on genetically modified tumor suppressor genes to understand how breast cancer arises specifically. It also discusses the importance of the tumor suppressor genes in itself. The tumor suppressor genes that were in question in this research study are P53, P10, RB1, and NF1. These tumor suppressor genes are very important. P53 is a gene that encodes for a protein which regulates the cell cycle and functions as a tumor suppressor. It is very important for cells in multicellular organisms to suppress cancer. P53 has been described as the guardian of the genome, referring to its role that mainly is conserving stability, which prevents genome mutation. This name is due to its molecular mass. It is in the 53 kilodalton fraction of the cell proteins. It also plays an important role in the cell cycle control and apoptosis. When P53 is defective, it allows abnormal cells to proliferate, which can result in cancer. As many as 50% of all human tumors contain P53 mutants. To talk about the second one, which is P10, this gene provides instructions for making an enzyme that is found in almost all tissues of the body. The enzyme acts as a tumor suppressor, which means that it helps regulate cell division by keeping cells from growing and dividing too rapidly or in an uncontrolled way. Also, P10 is linked to a lot of several mechanisms to this P53 tumor suppressor. Through P53 and additional mechanisms, the loss of P10 leads to genomic instability. P10 is so important because when it is defective or is lost, it misregulates multiple AKT-dependent and independent critical pathways for the development of cancer. Also, the RB1 gene, this is very important because it provides the instructions that make a protein called PRB. This protein acts as a tumor suppressor, which means that it regulates the cell growth and keeps the cells from dividing too fast or in an abnormal, uncontrolled way. In humans, the protein is encoded by the RB1 gene, which is located on the 13th chromosome. If both alleles of this gene are mutated early in life, the protein is inactivated and this results in development of cancer. And the way this protein got its name is because it results in retinoblastoma cancer. Neurofibromin acts as a tumor suppressor protein, which is the NF1. Tumor suppressors normally prevent cells from growing and dividing too rapidly in an uncontrolled and abnormal way. The NF1 protein appears to be very prevalent. Cell overgrowth is important because it prevents cell overgrowth by turning off another protein called RAS. And that stimulates the growth and the division. So to talk a little bit about more of the methods about this research study, they began the experiment by generating organoids from the basal and luminal progenitor cells, which were derived from normal breast tissue. So they had 12 donors. Following this, CRISPR-Cas9 was used to genetically modify the organoids of the 4 tumor suppressor genes which were involved in the formation of breast cancer. Researchers did most of their experiment in petri dishes, so they could be further analyzed, and so the organoids could be analyzed at a high magnification in order to see the cell's physical and characteristics and understand their chemical properties as well. So in the picture to the right, it represents organoids from the mature luminal progenitor and the basal subsets after 2 weeks. To further talk about CRISPR's role in this study, it was introduced into normal breast tissue where lentiviruses that had a specific genetic and physical makeup, they were injected. These lentivirus had single-guided RNA strands and were used to target 3 of the 4 tumor suppressor genes. The tumor suppressor genes that were targeted were P53, P10, and RB1. CRISPR was introduced in order to mimic neoplasia, which is also the abnormal cell growth and division, and it modified different combinations of inactivated tumor suppressor genes, which would trigger tumor growth, which is what they wanted. Netlin-3A was very important in this study because it activates the p53 pathway. So in this, in figure D, this represents the cultures that were exposed to Netlin-3A in order to select the organoids that were non-functional for the p53 tumor suppressor. And the image to the right, this represents an immunoblot analysis which was performed to determine the efficacy of p53, p10, and the RB1 of decreasing gene expression. The immunoblot analysis showed that p10 and RB1 were very highly effective in decreasing gene expression. However, what was shocking to the researchers was that the p53 levels did not decrease in the Netlin-3A selected cells, and their explanation for this was that it could have been due to a mutation of the p53 tumor suppressor gene that was not stabilized, and therefore this was not expected. So to talk about more about the efficacy of CRISPR-Cas9 in this research study, they performed a next-generation sequencing on organoids that were cultured in Netlin-3A. And this assessment revealed that luminal B breast cancer, which is classified as tumors that are positive for the estrogen receptor. And on this image, it is stated as ER as the top. And so also it is very important to note that in the image to the right, the 97 to 100% of the reads in each gene pool were mutated for p53 and p10 and RB1. To talk more about the analysis of the in vitro versus the mutated breast organoids, in the figure G, this describes the organoid proliferation, and they measured this by per week, and it was the multiplication per week. And as you can see, the proliferation rates of the control organoids did decrease over time. And their reasoning was that this could possibly be due to a loss of stem cell and possibly progenitor cells. In addition, the mutated organoids for p53, p10, and RB1, these did show an increase in proliferation relative to the control. And the researchers did expect this outcome. They had hypothesized this. Also, they did a second part of this experiment, and I believe that this was a very important, and this allowed for a lot of the information and for them to see whether their hypotheses and predictions were to come true or were to be supported. And for this next part of their experiment, they did most of these, they did this experiment within mice. So they did this to determine whether they could create tumors and whether this tumor formation was noticeable in mice. They also did this because they wanted to continue their hypotheses and predictions from the previous part of the experiment, which was mainly done in petri dishes. And this allowed them to really see the influence of deleting specific tumor suppressor genes and their combinations on tumorigenesis, because a lot of these tumor suppressor genes like p53, p10, RB1, and NF1 are critical whenever to suppressing tumors and to not allow for abnormal and uncontrolled cell growth. So as you can see from figure A down below, their process was that they used CRISPR-Cas9 to genetically modify the breast organoids, and they transplanted those into the mice, and then they assessed the mice for the tumor formation, and you can see on the far right that they did retransplantation, histology, sequencing, and drug testing. So you'll be able to see some of the results in the next slide. So in this slide, you can see the fluorescent imagery that was present, and these are just beautiful images. And they represent the fluorescent hyperplastic outgrowths, which were due to the transplantation of the p53, p10, and RB1 mutated organoids. And hyperplasia or a hyperplastic